I welcome you all to the lecture series for the A levels biology. In AS biology, we have to crack total of 11 chapters, and we will begin from the very first chapter, and this is entitled as the cell structures. Now, since the living organisms are to be bothered about, so you know well that the most of the living organisms they are complex, so they have these simple units in them, and these simple units they are referred to as the cell. We have got two major categories of the cells, like we have the eukaryotic and the prokaryotic cells. Like the cells of the kingdom Animalia, the plantae, the fungus, and the protoctus, they will have the eukaryotic cells, and the cells of the bacteria they would be the prokaryotic cells. There exist multiple differences, but as of now, you need to know and must remember that the eukaryotic cells have their genetic material which is enclosed by the nuclear membrane or the nuclear envelope, but the bacterial or the prokaryotic cells, their genetic material would not have a nuclear envelope or the membrane around it. Now, if I were to quickly take you toward that, how come the animal cell structure looks like? So this would have a living mass in it, and that living mass collectively comprises of the nucleus, the cell membrane, and the cytoplasm. And these three components collectively they make the protoplasm. If I were to tell you the further subdivisions of the nucleus, so there would be the presence of this nuclear membrane, there would come the nucleolus, then we will have the nuclear pores, then there would be the nucleoplasm, and then lastly we will have the chromatin within what? This nucleus. And then there would be the cell membrane, there would be the cytoplasm. Now, in close association with the nucleus, there would be a presence of a paired structure, which is a centriole, and the location where the centriole would be lying. So we would refer to this as the centrosome. Moving onward, we have got some membrane-bound organelles within the animal cell. They are entitled as the lysosomes. Then we have got something newly coming here in AS biology. This is termed as Golgi apparatus. And in the Golgi apparatus, further we have got two things to be talking about. One is the Golgi body, and then this is the Golgi vesicles. And collectively, they make an apparatus. The name of the apparatus is the Golgi apparatus. Further, we have got the small spherical masses in the animal cell, and these are the ribosomes. Then these ribosomes they can lie freely within the cytoplasm, or they would be adhered to or attached to the membranes, and these are the membranes of the rough endoplasmic reticulum. Then there would be the smooth endoplasmic reticulum, and then as overall for the human body, we have the muscles and the bones and the tendons and the ligament that make my structure of the body or that provides support to the body. So for the cell, we have got their intracellular skeleton, which is termed as the cytoskeleton, and the cytoskeleton is made up of the microtubule. Not all the eukaryotic cells, but most of the eukaryotic cells, they have got this last thing in them, and we refer to them as the microvilli. So let me quickly take you to be understanding the ultrastructure for an animal cell. So for the animal cell, you see that on the outside, what we have here, so this would be the cell membrane with us. Here we have the nucleus with us. Between the cell membrane and this nucleus, we have got this fluid-filled space here where most of the chemical reaction occurs that is indicated by the blue shading. So this is the cytoplasm. For the nucleus that we have got at the center, now there are further subdivisions. So in the subdivisions, you know that the nucleus will have this nuclear envelope, or this will have the nuclear membrane. And in the nuclear membrane or the nuclear envelope, we will have some pores in between them. And these pores right here, so these are the nuclear pores. Then at the center, we have got denser protoplasmic mass, and this is nucleolus. And then as for the cell, like there is existence of cytoplasm. So within the nucleus, there would be the presence of the nucleoplasm. Like with nucleus comes the word nucleoplasm. When the cell is not dividing, so there would be the existence of loosely arranged form genetic material. And this loosely arranged form genetic material that we have here, so this would be entitled as a chromatin, and the chromatin has the DNA molecules in it. When I would go on the outside, so in close association with the outer envelope of the nucleus, so you see that here we have got an extended network of the membrane. It goes like this, and if you were to see clearly, so this has got some kind of a rough appearance. The rough appearance is due to the fact that there would be the presence of these small spherical masses on them. These small spherical masses, they are the ribosomes, and they make the appearance of the rough endoplasmic reticulum as rough. So that is why this is named as the rough endoplasmic reticulum, we say. The ribosomes not only lie adhere to the membrane of the RER, but they could also lie freely within the cytoplasm, as you see here. So these again are the ribosome, but they are not the ribosomes of the RER, but rather they are the cytoplasmic ribosomes. Now, as is the case with the rough endoplasmic reticulum, now we have got some membranes lying here, and these membranes you see that they do not have a rough appearance, but rather they have a smooth appearance. And due to that smooth appearance, they are not termed as RER, but rather we call them as SER. S means smooth, and for the membrane we use the word endoplasmic reticulum. Moving further, if I were to take you close to the nucleus, so there would lie the paired structure, and these paired structure they are termed as the centrioles, and these centrioles they are present within an area, and that area we call this as centrosome. Now, there came a new word here a while ago that I was referring to as the Golgi apparatus. So, for the Golgi apparatus, you see that it further has got these membranes in them, and these membranes say are the Golgi bodies. And in close association with these Golgi bodies, we have got its own vesicles, and these vesicles they are entitled as the Golgi vesicles. So, these Golgi vesicles and the Golgi body they collectively make the Golgi apparatus. Now you must never be confused in between the configuration of the RER because RER is enclosed or is lying close or in attachment with the outer envelope of the nucleus. Okay, the SAR you see that compared to the Golgi body, they are a little bit more extended. They are a bit little of more larger. Okay, and they don't have any ribosomes attached with them. If you were to distinguish in between the appearance of the Golgi body with others, so you see that this Golgi apparatus or the Golgi body that we have, it has got two faces. Like on this goes like this. So you see, like this has got some appearance like this. And we will see later in our detail of the Golgi body that what is this face of the Golgi body and what is this face of the Golgi body. Now we can never forget about these structures that we have here. So these are the mitochondria, and as is the case with the mitochondria, so they have this outer membrane and they have got this inner membrane which we entitle as a cristae. So we have got this mitochondria and this lies here, and it could be present anywhere. Like it could be here. It is also the mitochondria. This also is mitochondria. This also is a mitochondria with us. And then here we have got something that are the membrane bound organelles, and they have got some digestive or the hydrolytic enzymes in them. So they are termed as the lysosomes. The thing doesn't end here, as I have mentioned, that as overall we have the bones and the muscles. So the cytoskeleton of the cell, the inner cytoskeleton of the cell, the intracellular skeleton that I'm highlighting with the yellowish marking, so they are the microtubules. Okay, not for all the eukaryotic cells, but most of the eukaryotic cells they would have these membrane bound structures, and these membrane bound structures we refer to them as a microvilli. That is a special feature of the cells that we have got in the trachea. Okay, like you may have learned in your IGCSEs that in the trachea there is existence of some ciliated cells. Okay, so the cilia and the microvilli they are something like that. They are the cytoplasmic, you could say not the cytoplasmic, but they are the extensions of the cytoplasm and they are enclosed by the membrane. So we refer to them as the microvilli. So this was overall the ultrastructure of an animal cell. Like I'm repeating it again, this is nucleus. This is rough endoplasmic reticulum. Here we have centriole. This is microtubule. This is mitochondria. This is lysosomes. These small yellowish kind of a thing available here. So these are the ribosomes. Here is the smooth endoplasmic reticulum. Here we have got the Golgi apparatus. Here is the cell membrane, then is the cytoplasm. So I hope you would be clear with the ultrastructure of an animal cell.